morning, everyone. My name is James Altland. I'm the research leader for the Application Technology Research Unit. And if you've been around the Maumee Valley Growers Association for a while, you know that we're a group of scientists that work for the U.S. Department of Agriculture. There's been a lot of changes with our organization in the last few years since the pandemic. We've grown a lot. And so what I wanted to do is 4 of us are going to speak today, and I'm going to be the first speaker. I'm going to give you a broad overview of our unit. Then 3 of our scientists are going to speak specifically about the type of research that they're doing and, how it relates to, greenhouse and nursery production in Ohio. So again, we are with the USDA. We're a branch of the USDA. And I show this slide, which is actually a little dated now, only to give you an idea of the scope of the research that USDA does. So we do everything from human nutrition, crop production, livestock production, food safety. And these units for the USDA are located all over the United States. Now, fortunately, each unit focuses on a different type of research. Fortunately, or at least fortunately for the Maumee Valley growers, the units located in Worcester and Toledo, we're focused on controlled environment agriculture or greenhouse and nursery production, as well as vertical farms. So now what has changed over the past, our numbers have grown. We have doubled in size. So these are the scientists that work for our unit in Worcester and Toledo. The ones that are highlighted in yellow, those are the scientists that are located physically on the campus of the University of Toledo. The rest of us, as Joe mentioned, are down at the Ohio State University campus in Worcester. Right now we have 10 scientists. 3 of us are horticulturists working in some facet of horticultural production. We have 3 agricultural engineers, and we're going to hire another one. This kind of blank face over here. We're supposed to be hiring another engineer probably next year, and that person will specialize in robotics and labor-saving robotics type research. We have 2 entomologists and 2 plant pathologists. So I'm going to give you kind of some background on all of these scientists, and what I'd like to tell our stakeholders is that some of the research projects that these people are working on, sometimes they're related to what you're doing and sometimes they're not, but these are all world-class scientists with incredible expertise in the type of production that you're doing. So if you understand what their expertise is, you can use them as a resource. And their specific research projects may or may not align with exactly what you're doing, but as I'll explain to you, they can still be an incredible resource for you as you come across problems or even opportunities in your business. So first up, this is me, James Altland. I'm a horticulturist and the research leader. Most of my research right now is focusing on the microbiome or the biological component of container substrates. So we're very good at measuring the chemical and physical properties. With new RNA and DNA sequencing techniques, we've only recently become capable of thoroughly studying the biological properties of container substrates. So we're really trying to map that out now and understand how that affects crop growth. We also do a lot of hydroponic vegetable production, and I do that with a postdoc down at Ohio State University. And she has expertise in hydroponics and aquaponics. So if fish are something you're interested in doing, that's something that we can help with as well. So she hopes to get out and share a lot of that research. Hopefully, if we can start to travel again this summer, you may see us at a few of the local seminars and grower meetings around the state. Dr. Jim Owen is another horticulturist down in Worcester. He's an expert in water. What are the qualities that water should have coming into the nursery or greenhouse, and how do we clean that water up before it leaves the nursery and greenhouse? Now, as it happens, studying water, you also have to be an expert in nutrition or the fertility aspect of it, as well as the substrates. So Jim really has expertise in all 3 of those areas and how they all work together. We have Dr. Marzano. Dr. Marzano is at the University of Toledo. She's one of our new scientists. She's a molecular biologist. She's doing some incredible research on using viruses to attack fungal pathogens in plants. And that's real timely now because of all this COVID. I mean, viruses are a hot topic right now. So I put Dr. Marzano under the umbrella of plant pathology because she is working in controlling plant pathogens. Although a lot of her work, I mean, she's working on a cellular and molecular level. So it's really high end, very, very sophisticated research, but it's all aimed at, oh, sorry, I apologize for that. It's all aimed at controlling diseases in greenhouse crops. She's shown some very good results with tomatoes so far. Dr. Testin is going to talk a little bit later to you directly. Anna is also a plant pathologist. You know, I think Anna describes herself as more as a classical plant pathologist. She does a lot of with disease diagnosis and cultural and chemical methods for controlling these diseases in high tunnels, greenhouses, vertical farms, etc. Mike Redding's one of our entomologists. He's a field entomologist. Based on his background and training, he's very good with all sorts of fruit trees and orchard crops. But he does a lot of work right now in wood boring beetles. And again, mostly from the field aspect, collecting samples from the field, identifying the beetles and developing control recommendations in the field. Chris Ranger is his partner in crime. He's another entomologist, but you know, working more on the molecular level in what triggers these insects to attack plants. What triggers an insect to do what it does in terms of attacking plants or not attacking plants. So he works very closely and complementary to Mike Redding in terms of, you know, Chris is more of the lab guy and where Mike is more the field guy. You all know Dr. Bolt, or most of you do if you've been around the Maumee Valley growers. She's really been the face of our Toledo lab for probably the past 8 or 9 years. Jennifer's going to talk more about her work, but she's a horticulturist working in greenhouse and vertical farm crop production. One of our new scientists in Toledo is Dr. Kale Harbic. He's an agriculture engineer, really a computer engineer. And again, he's going to talk a little bit later, but he works on the modeling of energy and light in greenhouse production. So very complementary to the work Jennifer does. Down in Worcester, we have Dr. Hong-Young Jun. He's a new scientist. He is an agriculture engineer focusing on spray technology. He has particular expertise in machine vision. How do computers see things, or robots? How do they see things and how do they interact with their environment? But again, he specializes in all aspects of spray technology, applying agrochemicals, not even necessarily pesticides, but also beneficial and biological type crop protectants. And all of you probably know Dr. Zhu. Dr. Hairping Zhu has been with us for a long time, has done a lot of spray technology research. He developed the intelligent sprayer that can move autonomously through a field and spray only where the plants are located. He's now adapting that technology for greenhouse crops. So you may hear a little bit about that over the next few years. Dr. Zhu and Dr. Joan work very closely together. Again, very complementary in the type of research they do. Very focused on spray technology and how to apply pesticides and other crop protection technologies more efficiently and more effectively. So again, I'm going to end here and turn it over to Jennifer, Dr. Bolt. But I just wanted to emphasize, these are the people that work with our unit. And again, some of their research may or may not align directly with what you're doing, but understand the expertise that these people have and use them as a resource. They will help you as much as they can. They have a job to do, which is research. And we're not extension agents, but if you've worked with Jennifer in the past, you know that a lot of these people, they like to get out in the field and meet with growers and talk about your problems or your issues or your opportunities. And they'll help as much as they can. But if you understand their area of expertise, you can utilize them more effectively as a resource. So unless there's any questions for me right now, I'm going to turn it over to Dr. Bolt, and she's going to tell us the type of research that she's working on.
Great, thanks, James. If anyone's got a question for James, feel free to ask that now while I get my screen shared. Okay. Joe's
looks good, Jennifer. Yeah, I'm gonna
okay.
Mute myself now and turn it over to you.
Okay, thanks, James. All right, so this morning I just wanted to give an update on some of our facilities developments, as well as some of the research that I've been working on over the past year or two. So first up, with the addition of Dr. Shini Marzano and Dr. Kale Harbic to our group, we needed to find some additional research space that is dedicated to the type of research that they're doing. So we've reconfigured our upstairs lab to accommodate Dr. Marzano, and down in the basement of Bowman Adi on the University of Toledo main campus, we have put together an engineering lab for Dr. Kale Harbic. He got to design it for his specifications, and he might speak a little bit more about that as he'll be presenting after myself. And then the second major update to our facilities is that we are in the process of developing 4 indoor growth rooms that'll be research scale vertical farm rooms. And these rooms are located at the Laboratory Incubation Center, which is on the health science campus. So the hospital is over here, and then just east is where our building is going to be. And so thanks to Google Maps, here's a outdoor view of the Laboratory Incubation Center. And then this is a blueprint of the inside. We've got 4 rooms here that are all a little over 400 square feet each that will be renovated. And then we'll also have some office space, and then what we're calling a prep lab. And that's where we can mix fertilizers, collect data, store samples, things of that nature. And so we will have almost half of this building dedicated to USDA ARS research space. And the nice thing about these 4 rooms that we'll have when they're fully renovated is that we'll have the ability to monitor all the environmental parameters in those rooms so that we can use that in quantifying how plants grow under different conditions. Also, we'll have sensors that will be able to be integrated with the HVAC system so that Kale, from the engineering side, will be able to look at energy usage, how much condensate is coming out of the system, and we'll be able to integrate components of plant growth with engineering capacity and looking at integrating that also with economics. So we started renovations here in late fall, in November, December, and so we're hopeful that come sometime this year, we will be able to move in and get that up and running. So to switch gears just slightly, for those that are familiar with me and my research and those that may not, I focus on the plant physiology side of controlled environment ag, which is a nice integration with Dr. Harbic's side of agricultural engineering. And so not a pop quiz this morning, but this equation is for photosynthesis. And so from my standpoint, looking at how carbon dioxide and light and temperature, water, nutrition, how all of that plays a role in plant growth and quality. And so the plants are able to utilize the carbon dioxide, water, and light to produce sugars and oxygen. And so then within the plant, these sugars can then be destined for 2 different fates. One is what we call primary metabolism, and that is just developing the leaves and the stems and the flowers and just developing biomass accumulation. but also the plants can direct some of those sugars towards what we call secondary metabolism. And that's the development of your flavor compounds, pigmentation for color, disease defense upregulation, and antioxidants. And so it's a balance within the plant, is that from that pool of sugars that they've been able to generate, how much of it is directed towards growth and how much of it is directed towards flavor. And then us as researchers, and then also for growers, finding out what balance of that is preferred by the end consumer, either for ornamental plants or for edible production. The project that I've been working on for about 2 years now is called Optimia. It's called Optimizing Indoor Agriculture for Leafy Green Production. And this is a specialty crop research initiative funded grant that's in collaboration with Michigan State, Ohio State, Purdue, and University of Arizona. And so overarching, we're looking at production, economics, and engineering for indoor leafy green production, but my aspect is on the production side. So this is a picture from one of the research projects in which we were growing the plants all with the same amount of light, but looking at different qualities of the light. So differing ratios of red light, blue light, and green light. And then we had a crop that was grown in the greenhouse as a comparison. And so what we can see here is that although they all got the same amount of light, they directed their photosynthates towards differing outcomes for morphology. So some plants had much larger growth, but a lot less pigmentation, whereas others had less growth, but more intense pigmentation. And so depending on the consumer, in terms of visual quality or taste, one might be preferred over the other, but it gives an idea of, depending on what your end goal is, you might pick a different light recipe dependent upon what you're looking for. And then from my aspect, I'm interested a lot in nutrition and how environmental conditions can impact nutrient uptake and how much fertilizer we need and where it gets allocated. So this is from that same study. We looked at the nutrient concentrations in the lettuce leaves. And to give you an idea, these first 5 are plants that were grown indoors in a vertical farm under differing amounts of light. So this one had, for example, just red light at 180 micromoles. And then this one had 20 micromoles of blue and 160 micromoles of red. And then we had a greenhouse comparison here. So we saw that for some of the elements that we were interested in, that the plants that were grown indoors had higher concentrations compared to the greenhouse grown lettuce. But that's not the whole story, because then conversely, there were other elements. For example, this graph is showing magnesium, but we had other elements that had higher accumulations under greenhouse conditions versus indoor conditions. So it's not a all for one kind of proposition. And so depending upon what elements might be maybe more important from a human health standpoint, or elements that you're looking to increase in order to have the ideal ratios within the plant, that could be better suited for indoor farms or for greenhouse production. And then another aspect that we've been looking at is modeling plant growth under various combinations of light and temperature and CO2. One, to try and see what is the ideal combination to get the maximum growth potential. But then also now we're starting to integrate that with resource usage to see what are the trade-offs if I want a desired rate of growth, would it be better for me to, say, add supplemental lighting, or maybe to look at increasing the CO2 concentration within the greenhouse? So we can take this slide here and transition it into a different outlay. So in this case, just to kind of set the stage for you, the plants were grown at 3 different temperatures, so 57, 70, and 82. And this is light intensity here on the x-axis, and then the CO2 concentration here. And the different numbers along each of these lines represents a different amount of growth. So one thing that we could look at is, say, if your desired rate of growth was 10, you could achieve it with a higher light intensity and a lower CO2 concentration. Or if it was a cloudier day and you had a lower light intensity, you could achieve that same rate of growth by having the greenhouse at a slightly higher CO2 concentration. And so we can then look to see along a different line, any combination of light and CO2 on that line would give us the same degree of growth. And then secondarily, we could say, well, okay, if we keep CO2 concentration the same at the lower temperature, you're going to need a much higher light intensity in order to get that same amount of growth. Whereas then at a higher temperature, then you would need less light. So it's looking at some of those trade-offs of, we know that electricity for lighting is expensive. We know there's also a cost for CO2 supplementation. We know there's a cost for heating. And so based on rates of growth, what would be compromise combinations that would give us the same growth, but maybe at a slightly lower cost point. And then lastly, I'll finish up here and say that we're just starting to dabble into another research project looking at strawberry propagation in controlled environments. So not on the fruit side, but for daughter plant production. And so we're looking to work with a team of researchers headed up by North Carolina State to look at ways that we can help produce high quality runners and daughter plants that are disease-free for then transplant out into field production or greenhouse or indoor fruit production. So with that, I will stop my screen share and I will turn it over to Kale. So, thank you.
Okay, can you guys see my screen?
I can, if you can just put it into presenter mode.
How's that?
Perfect.
Okay. So I'm Kale Harbic. I had a chance to meet some of you a couple years ago at the winter meeting in January 2020, but I'll just give a brief overview of some of my research projects. So as Jennifer mentioned, we have a new lab that just got finished being renovated, and it's looking pretty bare right now, but we're still moving in and setting up all our equipment. But we're excited to finally be able to move into that space. So we have a lot of plans for what we're going to do with it. I have a small team of engineering folks. And one of those positions is currently, we're still looking for someone to fill that position. But a couple engineering technicians and a programmer. So some projects we're planning to do from this lab are involved in light measurement and modeling. And I'll talk about that in a little bit. We're looking at some greenhouse drone development and control systems. So we are interested in greenhouse control systems and growth chambers, and we'll be able to build some custom hardware that deals with that in this type of facility. So we'll have a dark room shown on the bottom, which will be used in the light measurement project. We'll also be able to do 3D printing and CNC manufacturing for some of the projects. So the first project I'll mention is most of my work involves something to do with energy. So, we want to model the energy consumption of greenhouses and other similar facilities, and then see if we can find ways to optimize the control systems and save energy. So the first step is modeling. Virtual Grower is a project that Jennifer and I work on. We're hoping to have our next version out this year. So Yahya, our programmer, has been putting a lot of work in on that. I also do some GIS type stuff, geographical information systems, where we look at climate models across the whole country, and we look at the parameters for a typical greenhouse, and we try to see what are the supplemental light requirements based on different crops. So that's what's being shown on the graph on the right. We're also launching a new project on drones in greenhouses. So the prototype that we're building is shown in the upper picture. And if you can imagine a top-down view of a lettuce crop in the bottom picture, one of the things we'll be able to do with this platform is to do multispectral imaging. So we'll be able to look at infrared and ultraviolet light that is not visible to the human eye. And that could allow us potentially to do some early disease and water stress detection on the crop. And of course, we'll also be able to carry out surveys of the greenhouse in terms of, are there spots where we're getting good light or not so good light, and create a map of that environment. I also do a lot of work with lighting control and modeling. So the top picture is showing, the top left shows what sunlight looks like in a typical pattern. In the middle of the year, in the summer, you have high variation, but usually a lot of light at the peak. And of course, in the winter, you have very low light consistently. So can we take that variability in sunlight and use lighting and shade controls to make the light much more consistent, to have more consistent yields? So that's a control system application. And in the bottom picture, we're looking at, okay, if I have a bunch of lights in the greenhouse, how uniform is that supplemental light? Are there hot spots? What are some ways that we can make the light distribution more consistent, whether by changing where the lights are positioned or changing their relative intensity using better control systems? So the goal of both of those efforts is to ultimately save energy for growers and reduce energy costs and carbon footprint. And I'll wrap it up there and hand it over to Dr. Anna Testin.
All right. Good morning, everybody. Can everybody see my screen okay?
Hey, Anna, while you're getting that set up, I have a question for
Yep.
Kale. If Kale, can that drone do inventory? Can it count the number of plants in an area and maybe even grade the plants?
In the lettuce case that I showed, yes, it'd be able to do that. If it's a more heterogeneous environment with all kinds of different plants at different growth stages, that might be a trickier thing to do, but that's something we're looking at.
All right. Any other questions for Kale?
Yeah, this is Joe. Kale, real quick, can the drone also detect water content, whether it's inside the greenhouse or for our farmers outside within the fields?
I'm not looking at field applications specifically. There are some other ARS labs that look at that, but in the greenhouse case, if you were looking at multispectral imaging, some of our colleagues have shown that you can detect things like water stress from the plants using those imaging techniques. So it could theoretically be linked to a system that controls, when the plants need to be watered.
Thank you.
Okay, any other questions for Kale? Okay, otherwise, I guess I will wrap up our discussion of the new the research in the ATRU. So my name is Testin. I'm a research plant pathologist with ARS, and I've been in this position in Worcester for the last year and a half. And the overall goal of my position is to come up with disease management strategies for controlled environment agriculture. So we all know that's a very broad field with a lot of different crops and a lot of different production systems. And so the diversity of diseases and management strategies that I work with is quite large. Give my computer a second to wake up. There we go. So my background is very broad, which helps me in this position. So for my education, I did a tour of Big Ten universities, ranging from the University of Minnesota for my undergrad, Penn State for my master's, and Ohio State for my PhD. And in my time, I gained skills and research experience in disease management, disease diagnostics, biological control, and all of these have focused on specialty crop production. I've worked with native plants, specialty crops like quinoa, and most of the time I've worked with many different vegetable crops. I also gained experience in seed pathology while working as a plant pathologist at a USDA seed bank in Ames, Iowa. And in my research experiences, I gained experience in international agriculture, working in South America and Ecuador and Bolivia, and in Africa and Tanzania and Ethiopia. So no matter what disease I come encounter with, I can use all these past experiences to help me come up with practical solutions. So my lab is young and it's a little bit small. So currently I have a technician, Scott Shaw, who served in USDA ARS for 20 years. Then I have Margaret Mudispa, who's a lab assistant who's finishing up her master's at Ohio State. Last week, Manisha Hamill joined my lab, and this is a PhD student who's going to be co-advised with Sally Miller. So the overall goal of my program is to develop a portfolio of practical disease management tools for CEA producers. So in this research, we do emphasize coming up with better ways to diagnose diseases and monitor pathogens in these diverse systems. And to come up with better disease management strategies, some of our research does need to focus on fundamental aspects of pathogen biology. A lot of what we encounter in CEA systems are root diseases, such as diseases caused by Pythium in hydroponic systems, or soil-borne diseases in soil-based systems. Because of limitations on the use of synthetic pesticides that are established and also emerging regulations in a lot of these systems, we do focus on developing disease management strategies that don't rely on the use of synthetic pesticides. So a major area of research is looking at the use of anaerobic soil disinfestation to manage soil-borne diseases and soil-based high tunnel and greenhouse production. And Michelle Wallace will give a talk about that after my presentation. And I'll stick around for Michelle's talk if anybody has questions. So in this overall theme, we're looking at how we can manipulate the growing environment and its associated microbiome to create conditions that are less conducive to the development of diseases. So just to prime you for Michelle's talk, anaerobic soil disinfestation is a method in which we amend soil with a carbon source, such as wheat bran or molasses. So this acts as a food source for our native soil microbes. You then irrigate that soil to saturation, which fills all the water, all the pores in the soil with water, and then you tarp that soil for several weeks. So what happens in that time is that beneficial microbes in the soil break down that carbon source, and they create conditions that are really not conducive to the survival of soil-borne pathogens. So that soil goes anaerobic, and there's production of toxic byproducts such as organic acids like acetic acid, and also volatile organic compounds or gases that kill off soil-borne pathogens. So again, with disease management without synthetic pesticides, other areas of research in my lab include developing and evaluating biological control strategies available for growers. While biological control is not usually as effective as a synthetic pesticide, it is a tool that many of our growers are more reliant on. And lastly, we're looking at methods to produce clean seed or clean propagules using different technologies such as atmospheric cold plasma, so that growers are always starting with a clean slate in terms of diseases. So 2 large current projects that we have in the lab are optimizing anaerobic soil disinfestation for use in high tunnels. This is a project I actually started on during my PhD with Sally Miller. As part of this project, we're conducting a soil-borne disease survey in which we will test growers' soils in high tunnels or greenhouses for free, and we'll provide the growers with a report of soil-borne diseases. So if that's something you're interested in, please contact me. We're also looking at a project to evaluate how different biostimulants could potentially reduce diseases in hydroponic systems, with the idea here being that if we add a specific biostimulant, this will change the microbiome of these crops and hopefully reduce disease. So my program also really focuses on meeting the needs of growers and my stakeholders through effective communication and engagement. And unfortunately, the pandemic has made it really difficult to meet with a lot of the growers. I know, luckily, Scott attended the workshop that I did with Michelle Wallace this past summer. So it was really good to meet some growers. But really, I would love if you could reach out and let me know what your disease management needs are. I really enjoy talking about plant diseases, so feel free to reach out by email or phone to let me know what your biggest challenges are. And then again, if you are producing in either a high tunnel or a soil-based greenhouse, we do have this free soil-borne disease testing service that's being sponsored by my lab and Dr. Miller's lab. So please reach out if you're interested in that service. And that was all I had for today. So if anybody has questions or comments, please let me know.
We have a few minutes, so those of you that would like to bring up some questions or concerns or describe some of the challenges that you've had in the last year or two, this is a great time to do that. We have USDA scheduled until quarter after. That's 12 more minutes or 7 more minutes from now. So let's take advantage of it. Unmute first.
Hi, Joe. Mark Langen here. Could I get contact information for James Altman and Jennifer Bolt?
Sure, no problem. I'm making a note of it now. I'll send it. I'll email it to you. So you want Jennifer and Dr. Altman, okay?
Hey, Joe, I'll also mention right now, USDA ARS is not allowed to travel. Ohio State University scientists I know can, but ARS, and this is not my decision or the scientist's decision. It comes straight, from the White House. Now we are allowed to meet with stakeholders, but we can't go to, big meetings or conferences and things like that. So there are some restrictions on how we can travel and what we can do. But, I know myself and some of our scientists, we bend the rules as much as we can in order to get out and meet with the growers. So if there's a need there, please reach out to the individual that you're interested in, talking with, and we'll do what we can to accommodate you. Although just, officially, we are still not supposed to travel. So I just want to put that out there and let everyone know what the current regulation is for us. And that's all because of the pandemic, of course.
And for everyone on the call today, I put my email address in the chat box.
Yeah, maybe James can do the same thing too.
I will do that for sure.
Thank you. Jennifer, you and your peers, how long is the funding available roughly for the projects that you're working on right now?
Well, I will say that the majority of our research is appropriated funds. So Congress dictates our levels of funding from year to year. And so that funds the majority of our research. And then we do apply for supplemental grants to help augment the research that's funded through USDA. So right now I'm on a few grants, one with Kale to look at lighting, and that grant is for another year of funding. The Leafy Green Project grant has 2 more years of funding, and then the Strawberry Grant that was just funded this fall, so that'll be a 4-year project.
Thank you.